It's my great pleasure to introduce my friend Eric Verdin, who runs the Buck Institute, which, when I was at Sense Research Foundation, we used to have a little joke that if you wanted to read next year's annual report from the Buck Institute, you just needed to read this year's annual report from Sense Research Foundation, but that was completely unfair. The Buck Institute does wonderful work, and I'm delighted that the person who runs it has been able to join us online and talk about what's going on. He does seem to have a technical issue right now.
Can you hear me?
Ah, that's better. Yeah, there you are. Can you hear me?
My apologies. Good to see you, Aubrey.
Yes, absolutely. No problem at all. I guess you're ready to share your screen or get going. But anyway, I've just introduced you already when you couldn't hear me, so I've done all that. Right.
All right, super. Let me share my screen and first start by thanking you and Martin for the opportunity to be at this meeting. I apologize for not being there in person. I just could not make it work from a logistic point of view. Can you see my slide?
Yeah, we can see them.
I'm not sure. I did not hear.
Yes. Sorry, Eric.
Okay.
Yes, we can see your slides. It's working perfectly.
All right. Okay, perfect. Thank you. So just wanted this today spend some time describing new work in my lab, which is not a finished story, but I think it's a clear direction in which we're going. About 4 years ago, we started an increased focus in my lab on the biology of immune aging, in particular, focus on the human immune system. And I'm showing you here a number of key people in this new group, and I will be bringing them up as we go through the presentations. Just a way of introduction, I really believe that blood cells represent a fascinating window into human aging. And I want to introduce the subject very briefly for those of you who do not have an immunology background. So almost all blood cells are derived from a common precursor, the hematopoietic stem cell, which divides in two broad populations. On the right, the myeloid progenitors, which will give rise to most of the innate immune system. So this is an immune system that responds directly after an infection or an insult. One of the cells that many of you are familiar with are the monocytes or macrophages. The other arm of the immune system, a so-called adaptive immune system, is the one that is able to be educated as it encounters a variety of antigens and forms the basis for what we call immunological memory. And it is comprised mostly of B cells that I'm showing you here, and T cells, which each have their own progenitors. And these cells will differentiate into a large number of effectors, which are most of them actually present in the blood. Now, one of the hallmarks of aging is the fact that the thymus, which is the organ where most T cells will become educated, shows a dramatic involution during aging. Just the thymus, if you're not aware, is a small gland right behind the sternum. And what I'm showing you here is what happens during aging. You can see that by age 40 to 50, most of the cortex and the medulla, which are the T cells themselves, have been replaced by fat. Therefore, there's from age 40 to 50, one relies mostly on the T cells that have been developed and educated during the early part of life. And there's therefore a limit there in terms of what we can expect in terms of maintaining a functional immune T cell-based response. Now, just a few words also, because this will be coming back throughout my presentations. The T cells themselves, so naive T cells shown on the left, originate in the thymus. And again, most of these T cells are generated early in life until age 45 to 50. Will, as they encounter their antigens, will undergo a step of discrete differentiation steps that are shown here. They will become effector and early memory, eventually central memory, effector memory, and the final differentiated cell state called TEMRA. As these cells progress through their differentiation, they acquire a number of different properties. So they go from a very stem-like type of cells that are highly proliferative, that mostly utilize lipid metabolism, to, on the right, cells that have been called senescent, and this is something that we will be discussing together, that show increased cytotoxicity and a more glycolytic metabolism. So one of the reasons we decided to focus on this whole area of human biology is first the realization that looking at blood cells in some way might be recapitulating the whole process of aging. Obviously, the blood is highly accessible and therefore allows us to follow humans. And this might become really critically important in the future as we think about interventions and therapies or lifestyle factors that might be modifying the aging process in humans. Now, major reason we think this subject is of high importance is the fact that and I'm showing you two papers that were published in the last 3 years, highlighting the fact that immune aging by itself is interesting as an organ that ages, and we can measure this, but also has secondary consequences on an organismal level. So what both of these studies showed is that if you modify T cells only, and it was done on the left paper by deleting a critical factor only in CD8 T cells. This factor is TFAM. This is a factor that is a critical transcription factor for mitochondria. And so these animals had defective, basically, CD8 T cells and showed accelerated aging. And this is shown by a variety of markers. These mice actually have also significantly shortened lifespan. On the right side, this is a paper by Laura Niederhofer and colleagues. They used, again, a lymphoid precursor, promoter, to delete a gene called ERCC1, which is a critical protein participating in DNA repair. And so these cells also show intrinsic accelerated aging, but more importantly, in both of these papers show that when you affect the ability of T cells and accelerate their aging, you actually see organismal aging. And on the right side, I want to point to a variety of organs in which P21 and P16, two markers of senescence, were shown to increase. So the key message of this slide is that intrinsic aging is important. Obviously, you need to have a functional immune system to age well, but also it has secondary consequences at the level of multiple organs, and therefore could become itself a driver for aging in an organism. So the first series of experiments that I'm going to show you were focused on revisiting this whole thing. And in particular, so we used a number of humans in this particular experiment, 46 humans, aged 20 to 82 years old. They were of various CMV serostatus. And so CMV, again, is a virus that is very prevalent in the human population, and in some studies has been shown to correlate with aging. And so when you study immune aging, it's always important to include this as a variable. And our groups here were equally divided in some patients that were seropositive and some seronegatives. Now, the thing I want to highlight here is a new technology that we have used to revisit the whole process of immune aging. It is a form of flow cytometry with much higher resolution. So your traditional flow cytometry would typically stain 6 to 8 colors. In this case, this new technology called spectral flow cytometry can use up to 38 different colors, actually even more, but in this case we use 38. to really deep immunophenotype individuals, and really bring up a whole level of resolution. And I'm showing you on this next slide the type of spectral analysis that we can conduct. Now, there are some complex algorithms associated with this machine that essentially allows you to identify each of these fluorescent markers and to ascribe the antibody to which they correspond, and therefore to quantify different populations. And an example of this is if we apply this to whole blood. On the left side, I'm showing you what we call the dimensionality reduction plot, which essentially takes all of these variables and allows you to now identify a whole series of different cell subtypes that you can see here, including your monocytes, your CD8 T cells, your B cells, your NK cells, and so on. And we can now then utilize our patients to look at what happens to each of these cell types during aging. You can see on the right panels that cells change during the aging process. Your NK cells tend to increase, your monocytes tend to increase. The increase in monocyte is something that's been long described. It's part of what is generally referred to as inflammaging. But remarkably also, we find as humans age, a decrease in CD8 T cells and in gamma delta T cells and other specific subsets. Now, this is something that's been, this is not our unique discovery. There is a known shift from myeloid to lymphoid to myeloid type of changes that occur during aging. We can also do the same type of analysis at a much higher resolution, for example, focusing on the CD8 T cells alone. And again, here, using this type of tool, we can, on the left side, using the UMAP type of analysis, we can now identify a large number of different CD8 T cell subset, many more than actually have been previously identified. And interestingly, and I won't have the time to go through this today because these results, these experiments are ongoing, we've actually identified some new subsets that have not been identified before that are specifically increasing during aging. That being said, I think I want to now point you to the right side to show you what happens to the naive CD8 T cells, and you can see the dramatic decrease that occurs during aging. Again, this is a reflection of the fact that we have a defined subset of naive T cell that is generated in the thymus, and then once the thymus is gone, these cells basically progressively decrease as a function of aging. We can also see that the opposite occurs for the other CD8 T cells that are further on their differentiation path. For example, your central memory T cells actually increase. There are two different cell subtypes here called CXCR3 minus and CXCR3 positive, and your effector memory T cells. So here again, we see a shift in the population, which is quite dramatic. Now, the other variables that we have focused on during these studies is trying to use some of the tools to identify what is generally referred as senescent T cells. And I want to bring up here a technique that was pioneered by Judy Campisi at the Buck Institute, where in 1995, this was the landmark paper where she identified she was able to identify senescent human cells in the skin and other organs using a unique marker. And this marker is shown on the left. This is a precursor called XGAL. It is a hybrid between galactose and an indigo monomer. Now, this precursor gets cleaved by an enzyme called beta-galactosidase, or beta-gal, as I will refer to later in this talk. And once the indigo monomer is freed, it becomes colored, and one can actually detect it into a variety of cells. And I'm showing you a classical picture on the right. So this is something that's been used in many experiments, and is generally assumed to be a specific marker of senescence. And some of my presentation will be trying to determine whether this is actually true or not. And I just a forewarning. I think it seems that it is a lot more complicated than what we had anticipated in the past. Now, the beauty of this essay is that a number of scientists have generated additional markers that actually can assess beta-galactosidase activity. And I'm showing you here one called FDG, which is of particular interest to us. So again, you have a galactose molecule, in this case, not linked to indigo, but linked to a FITC, which is a fluorescent marker. So when this molecule gets cleaved by beta-galactosidase, it liberates the FITC, which becomes a bright green fluorescence, which we can then assess using flow cytometry. And there have been, so the reason we became interested in this is that there have been a number of studies using this type of marker on peripheral blood mononuclear cells, claiming that it reflected senescence. And again, the point I'm going to try to make in the next few minutes is that the picture is partially correct, but it might be more complicated than what we had anticipated. And in some way, I hope that this would serve as somewhat of a warning to the blind use of beta-gal and FDG as a marker for senescence. So the first experiment that we did is to, using again the spectral flow cytometry that I've shown, combined with a whole series of markers for CD8 T cells, and try to determine whether this beta-gal activity in T cell increases. And indeed, what we find is that when we go from naive T cells to central memory T cells to effector memory T cells, we are seeing a dramatic increase in the staining with beta-gal. So the y-axis here is beta-gal. The x-axis is the CD4 T cells on the left, the CD8 T cells on the right. And what you can see when we compare young and old donors, the cutoff was around 50 years old. You can see that we see a very strong increase in the number of beta-gal positive cells, shown here in red. Now, one of the things that has disturbed me and others in the field, and so there are a number of papers claiming that these cells are indeed senescent, but there's one variable that is of concern, is that you can see, if you look at the y-axis, that the fraction of these cells that are SA beta-gal positive can rise as high as 40%. And on average, it's close to 15 to 20%. Now, this is a number that is not consistent with what we see in terms of senescence in other organs. In most organs, even during old age, the fraction of senescent cells rarely rises above a few percent. And here in this case, we have some donors that showed 40% SA beta-gal positive cells. So the question that we've been interested in is, first, is characterizing these cells at a molecular level and trying to understand what are we truly measuring with beta-gal in the immune system. And so the first experiment we did that I'm going to show you is to essentially sort them. The beauty is of these techniques is using flow cytometry, we can not only identify cells, but we can also separate them and isolate them to some degree of purity. And so the first thing we did was to isolate these beta-gal positive CD8 T cells and to conduct an RNA-seq analysis to try to look at their patterns of gene expression. And I want to bring up another study that was published by Sandeep Khosla and colleagues at the Mayo Clinic, where they identified a new gene set that is apparently really closely associated with senescence. And so this data was originally obtained from young versus old human bone biopsies. They also found the same genes were upregulated in bones of aged mice. This was validated across multiple studies and tissues. And the net result is they identified 125 core upregulated genes that are heavily biased towards cytokines, chemokine, and their receptors, and tissue remodeling factors. So we were able then to take our pattern of gene expression and to compare it. Compare our genes that were upregulated in beta-gal high cells and those that were present upregulated in the SenMayo data set. And I'll ask you to focus on the right side, where we're looking on 4 different subpopulations: the CD4 central memory, CD4 effector memory, CD8 central memory, and CD8 effector memory. And the yellow color here indicates those genes that were upregulated in beta-gal in comparison to high versus beta-gal low. And what you can see is, particularly in CD4 T cells, is a very strong correlation between the genes that were present in SenMayo and those genes that are upregulated in beta-gal high cells. Now, that correlation, and we actually identified 52 genes out of the whole, out of the 120 genes from SenMayo that were really concordant between the 2 phenotypes, suggesting that indeed, at least in CD4 T cells and in CD8 T cell central memory, there is a gene expression pattern which is consistent with senescence. And the probability that this would occur by chance, as you can see here, is extremely low, at 10 to the minus 12. I should point also to the fact that the CD8 effector memory T cells, which are the most differentiated T cells, the correlation was significantly less strong. Now, one of the hallmarks of senescence is the fact that the cells actually stop dividing and become resistant to apoptosis. This is something that's been defined by Judy and her group. And so we were interested in trying to determine whether these beta-gal high CD8 T cells indeed showed decreased division or not. And we found something quite interesting, because when you look on the left panel, this is a dilution plot. Every peak corresponds to 1 cell division. And you can see that the SA beta-gal low and high cells actually divide pretty much to the same extent. And this is true when they are grown on their own. However, when we mix them with other blood cells, we found a very specific defect in cell division. You can see a very sort of a right side peak for the red cells, and which means the cells actually stop dividing, versus the blue, the upper panel in blue, showing that the say beta-gal low have actually divided to a normal extent. So there is in these cells, in these SA beta-gal high cells, a cell non-autonomous defect in cell division. Now, we're in the process of trying to identify the mechanism for this, but it would be clearly something that is distinct from normal senescent cells. Now, I'm going to show you another number of experiments that we've shown that sort of brings up some additional information and color in terms of what these SA beta-gal cells are. We found that when we compare SA beta-gal low and high, again on the left, that the low cells express low level of lysosomal tracker. So these are a marker specific for lysosome. But we also found that in the high cells, a very strong correlation from a morphological standpoint between lysotracker and SA beta-gal. Indeed, beta-gal seems to be marking lysosomes, and these actually dramatically increase during aging. Now, we find the same thing using another marker, in this case P62 and LC3, which are 2 markers that are associated with autophagy. And you can see again on the bottom panels, the SA beta-gal high cells show a dramatic increase in P62 and in LC3. Again, consistent with a decrease in autophagy. And, obviously, for the aficionados, there's a clear link between autophagy and lysosomal function. Finally, we also found that these SA beta gal high cells show another marker that is generally associated with senescence, which is an increase in gamma H2AX. It's not a specific marker, but it can be associated with senescence. And in particular, you will notice that on the bottom right panel, that these gamma H2AX staining is present mostly in the cytoplasm. Now, given the fact that this is a histone protein, this is consistent with another hallmark of senescence, which is the so-called cytoplasmic chromatin fragment. During senescence, there's evidence for the extrusion of DNA out of the nucleus. And in this case, we can directly see this by the presence of this histone protein associated with these cytoplasmic granules. Now, another interesting link that we have made is with lipofuscin. This was work that was supported by Michael Grieve and the Forever Healthy Foundation, who were interested in understanding whether what is the role of lipofuscin in this process. Now, lipofuscin is a complex mixture of oxidized protein and lipid degradation product that has been known to be associated with aging. I won't have the time to go through all of the background literature. There's obviously, as these represent sort of cross-linked lipid and protein product, they accumulate during aging, and there's therefore thinking that they might themselves become a part of the problem associated with aging and accelerate aging themselves. So using the flow cytometry, we are able to actually detect lipofuscin autofluorescence. Again, we doing work with Herb Kassler, we were able to identify a somewhat narrow band between 525 and 680 nanomolar that corresponds to lipofuscin autofluorescence, and we have a whole series of experiment that actually proved this case. I won't have the time to go through them, but what we found is that indeed it increases with age in T cells. Again, we're comparing young and old CD8 T cells, and you can see that there's obviously a very strong increase in these cells in red as the cells become progressively differentiated. And this is much increased in aging. This is the red versus the blue. And the numbers, frankly, actually closely correspond to what we had seen in terms of SA beta gal. And we have actually other evidence that there's a strong correlation between beta gal, between lysosomal staining, as I've shown you in the first few slides, and lipofuscin autofluorescence. And in some way, we believe that all of those represent markers of lysosomal stress. Now, finally, a functional experiment that we have done, and we've done this a number of times, is try to look at what is the functional consequences of these SA beta gal high cells. What do they do? Are they toxic? Are they, do they have any secondary consequences? And in this experiment, I'm showing you an early experiment, which we are very excited about, which shows that if we take the conditioned medium from these SA beta gal high cells, we can actually induce significant toxicity in primary human endothelial cells. So these are endothelial cells from humans that are cultured, and we can simply take the supernatants from the SA beta gal low or high. And on the right panel, you see the quantification that the conditioned medium from the SA beta gal high cells actually is remarkably toxic. Now, we're in the process of trying to identify the factors that are secreted by these SA beta gal high cells that actually leads to this toxicity. But this could be, we believe this is a sort of a molecular example of the point that was made by Laura Niedernhofer and others, showing that T cells by themselves age, but also have profound consequences on secondary cells. So this is a system that I think will allow us to really get to the bottom of what the mechanism might be for this secondary effect. Now, I want to finish by highlighting what our current model is of what we are measuring. So I'm starting with the top cells, which would be your normal T cells, which is, we know when these cells become activated, we suppress autophagy. These cells eventually on the right side will normally proliferate, and this will eventually dilute whatever lysosomal aggregate might be occurring. Now, during aging, or during genotoxic insult or negative co-stimulation, something happens. These cells stop proliferating. They undergo strong lysosomal stress, and a process that is looking like senescence, but it's not exactly senescence. As I mentioned, there are a number of key differences between a traditional senescent cell and what we observe in the immune system. And so now we think that the accumulation of these cells could play a significant role in inflammation and could be really leading to some of what has been observed, the secondary toxicity of T cells. Now, I want to finish. Obviously, this is a work in progress. We don't have all the answer, but we are continuing to work on this. We're quite excited by this new line of investigation. I want to finish by, again, thanking the supporters of this work, Forever Healthy and the Michael Antonoff Foundation, the NIH, and highlight also really the key people in my group who have contributed to this. Herb Kassler, for whom I don't have a picture here, but has been working with me for the last 20 years, is a bona fide card-carrying immunologist, has been instrumental in not only in this work, but also in establishing a great flow cytometry core for the whole BUC community. I also want to highlight Marlene Cervantes, Nicola Andrews, Hiroyuki Matsui, Ariel Floro, Alan Tomusiak, Christina Alexandru, and Ritesh Tiwari, who were a key part of this project. And I will stop here, and I'll be happy to answer your questions.
Great. Thanks so much, Eric. Very, very exciting findings. We have time for, well, at least one question. Anyone? Here's George. Go ahead.
Yeah, thank you. The process you have been describing, are they very exclusive to the CD8 T cells, or are they also affecting other T cells, B cells, and so on? And could there be more paracrine enforcement of this process, of this leading to this exhaustion and other problems?
So it's an excellent question. We have focused actually on CD4 T cells and CD8 T cells. The B cells have a very different sort of a development. They do not rely on the thymus, so there's not that barrier to thymus development or thymus involution. But there is other processes that occur associated with B cells, including the development of these age-associated B cells, and this is something that we are working on as well, and particularly in the context of mitochondrial biology.
Any other questions? One over here.
Great talk. I was intrigued by the gamma H2X in the cytoplasm. Have you looked at the, obviously it's a large structure to move out, have you looked at nuclear envelope integrity? And
yes, it's a very good question. We did, I did not show you the slide, but we find, consistent with what has been shown in other systems, that lamin B, for example, is lost in these SA beta gal high cells. This is a process that's been referred to as delamination. It is thought to be one of the sort of the hallmarks associated with senescence, and we do indeed find a delamination in the T cells that are SA beta gal high.
Yeah. Have you ever looked at the protein barrier to autointegration factor 1, BAF1 or BAMF1? It's part of this, it sits on the nuclear envelope and it's part of CGAS STING pathway as well as DNA repair.
No, we haven't, but that sounds like a great experiment for us to do.
Okay, thanks, Eric. That was really great. Another round of applause, please.
Thank you again, Aubrey, and it's good to see you all.